Mendel's law of inheritance. In this module, we'll discuss about what is inheritance, history of genetics, the 7 characters studied by Mendel during his experiment, the 3 laws of inheritance, reason for Mendel's work success, what is Punnett square, incomplete dominance and codominance. So let's get started. What is inheritance? Inheritance is the transmission of characters from the parents to their offsprings generation after generation. These offsprings they get all the genetic materials from the parents. Therefore, inheritance explains why the parents and their children have a similar characteristic traits. These characteristic traits which are passed on by the parents it includes for example the color of the eye, hair, skin, blood type, height, etc. Even diseases like Down syndrome, thalassemia can be passed on to the offsprings from their parents. Humans have 23 pair of chromosomes, out of which 22 pairs are autosomes and 1 pair is an allosome. In humans, the eye color is an example of an inherited characteristics as shown in the picture. An individual might inherit the brown eye trait from one of the parents. Inherited traits are controlled by genes, and the complete set of genes within an organism's genome is termed as its genotype. History of Mendel. Gregor Mendel was the person who worked on the discovery of basic principles of genetics. In 1866, he was the first person to shed light on the way in which characteristics are passed from generation to generation. He was a monk and he used to teach natural sciences in the monastery school in Brunn, Austria. He presented his data and conclusions in the paper Experiments in Plant Hybridization in the year 1856. He is known as father of genetics for his tremendous work. In 1902, 3 scientists, Karl Karen, Germany, Hugo de Reis, Netherlands, and Erich Mark, Austria, rediscovered his work independently. And after their rediscovery, it was named as Mendelism or Mendelian Law of Inheritance. Mendel's experiment. Mendel observed and selected 7 different contrasting characters in a pea plant, and each of these characteristics had 2 forms, one dominant and other recessive. The 7 pairs of contrasting characters were present on different chromosome pairs and were able to assort independently. The characteristic includes the first, as shown in the picture, the seed shape, which was round and wrinkled. Here, the round was the dominant character and wrinkled was the recessive one. The second character was the seed color, which was yellow and green. Yellow was the dominant one and green was the recessive. The third character was the flower color, as white and violet. The violet one was the dominant one and white was the recessive one. The fourth character was the pod shape. The pod shape was inflated and constricted was taken. Here the inflated one was the dominant one and constricted was the recessive. The fifth character was the pod color, which was yellow and green. Here the green one was the dominant one and yellow was the recessive. The sixth character was the flower position or place, we can say. It was in the axil and in the terminal pattern. The axil was the dominant pattern and terminal was the recessive one. And last character was the stem size or stem height, we can say. It was in the pattern of tall and dwarf or long and short, we can say. The tall was the dominant character and the dwarf one was the recessive one. Mendel's laws. Based on the theoretical observations and explanations of the experiments on garden pea, Mendel drew some important conclusions. These conclusions are regarded as Mendel's law of inheritance. These laws were as follows. The first, law of dominance. Second, law of segregation, which was also termed as law of purity of gametes. And lastly, the third law was law of independent assortment. So let's discuss these three types of laws one by one. First, law of dominance. Mendel's first experiment were with the varieties of garden pea that differed in only one visible character. According to this law, if a cross is made between plants with contrasting pair of characters, the character that appears in the first generation is dominant and the other is recessive. For example, when Mendel crossed the true breeding red flower pea with the true breeding white flowered pea, then in the F1 generation, only red flowers were formed. It was immaterial whether this red flower which was formed, it was a male parent or a female parent, which means it was not clear that the flower contains a pollen in it or an ovule. The second law was law of segregation, which is also termed as law of purity of gametes or law of splitting of hybrids. According to this law, if a cross is made between plants with a contrasting pair of characters, then both the characters are present in hybrid of F1 generation, where one is dominant and other one is recessive. The dominant character is expressive while the recessive one is hidden or which is not expressed properly. For example, if a cross is made between a red pure flower and a white pure flower, the red flower is denoted by capital R, and the white flower is denoted by small r, as shown in the picture. Then all the plants of F1 generation are red colored as shown here. Then if now these plants are self-fertilized and there is reappearance of white flower in the F2, then while the F1 plants had only red plants, it means that F1 plants also carry some hereditary factors for white flowers which is transmitted through red flowered F1 plant, unaffected by its close association with the factors of redness. This suggests that the parental trait, that is the factors, didn't mix with each other in F1 hybrid but simply lived together without affecting each other. When F1 plants make a gamete factor of allelic pair, they segregate, that means they separate from each other, and the gamete receives only one factor of a pair. Thus, the gamete carries only one of the two alternating factors, either for the red or for the white color. This is known as law of segregation, which means when a pair of allelomorphs are brought together in the hybrid, they remain together in the hybrid without blending but segregate completely and pure during the gamete formation. The phenotypic ratio which will be observed, which will be obtained in the law of segregation is in the ratio of 3:1, where we are going to get 3 red colored flowers and 1 white colored flower. The third law was law of independent assortment, which is also named as law of free recombination. Mendel studied a cross which involves two pairs of contrasting characters. First, round and yellow-seeded homozygous plants, which is denoted by capital Y, capital R, and second one was wrinkled and green-seeded homozygous plant, which is denoted by small y, small r. The round shape and yellow color of the seeds are dominant over wrinkled shape and green color. During gamete formation, each pair of alleles, they segregate independently of each other. Thus, the round and yellow-seeded plants produce only capital R, capital Y gametes, not capital R or capital Y. Similarly, the wrinkled green-seeded plants, they form only small r, small y gametes. All the plants produced in F1 generation were with round and yellow seeds as capital Y, capital R, small r. That means they are heterozygous for both the alleles and so they are called as dihybrid. In F2 generation, the F1 hybrid produces four kinds of gametes in equal number as capital Y, capital R, small y, capital Y, small r, and small y, small r. All these are four kinds of pollen grains and four kinds of egg cells. There are chances of 16 possible combinations among the gametes. But the union of these gametes is random and leads to the formation of 9 classes of genotypes and 4 classes of phenotypes. The 4 classes of phenotypes are first, round yellow, which is 9 in number. Second, wrinkled yellow, which is 3 in number. Third, round and green, which is again 3 in number. And the last one was the wrinkled and green, which was only one pair is present there. The plants with round yellow and wrinkled green seeds are parental combinations, but round green and wrinkled yellow are new combinations. The ratio of F2 generation, that is 9:3:3:1, is called dihybrid cross. This above result, it shows that each pair of alleles strictly segregate and is independent of each other, which demonstrates the law of independent assortment. Reason for the success of the Mendel's work. Firstly, Mendel probably chose to study the peas, that is Pisum sativum. The reason for his selection was they were easy to grow and they were available in many readily distinguishable varieties. Secondly, he kept accurate pedigree records of several genes so that the ancestry of the given plant could be traced back to the beginning of the experiment. Third, Mendel confined his attention to one character at a time. When inheritance of one character was properly established, then only he used to study the inheritance of other characters. Fourth, he was lucky in the choice of the garden pea. In this plant, the self-pollination and fertilization is possible. The artificial cross-pollination can be also done very easily. Fifth, the 7 characters which were studied by him in the garden pea, they are located on 4 different chromosomes. And those which are on the same chromosome, they are very far from each other. The law of independent assortment was possible due to this reason only. And lastly, the reason can be taken that he made quantitative studies of each progeny, which resulted from a cross, and his experiments dealt with a large sample size. Punnett square. So let's discuss what is Punnett square. Punnett square is a checkerboard which was devised by an English scientist named as R.C. Punnett in the year 1906. It is a geometrical device, a square with vertical and horizontal columns. The possible types of female gametes from the female parent and male gametes from the male parents are shown in the small circles above the vertical column and also at the right side of the horizontal column, as shown in the picture. The possible genotypic combinations and their phenotypes are then entered in the boxes of the square. Now let's discuss how we can draw this Punnett square. For this, I am taking an example of an monohybrid cross. Step 1, draw the 4 boxes. Step 2, note down the alleles in gamete from each sex, that is male from the male and the female. Put the female gamete horizontally along the top and male gamete along the left side. Step 3, note down the products of fusion of these gametes in the appropriate boxes. And lastly, the step 4 will be we are going to note down the phenotypes and genotypes of the offsprings. Incomplete dominance. Mendel's law of dominance states that one allelomorph dominates over the other, and as such, F1 exhibits one of the two alternative phenotypes present in the parents. However, there are some characters which are exception to this rule and show incomplete or a partial dominance. In such cases, the dominance is absent and the hybrid individuals they resemble neither parents and are intermediate between the two parents. This phenomena is known as incomplete dominance. The phenomena of incomplete dominance can be explained on the basis of Mendelism segregation law. In incomplete dominance, both alleles have almost equal effect on the phenotype, which results into an intermediate character of the hybrid. For example, as shown in the picture, is an example of an animal here. Here we have done a cross between a chestnut brown colored horse with a creamy white horse. And in F1 generation, we'll be getting a palomino horse colored here. Another example can be taken as from the flower plants. For example, when a cross is made between a true breeding red flower plant as shown in the picture as capital R, and true breeding white flowered plant as small r, then F1 will be your capital R small r where all are pink colored flowers. When F1 pink flowers are self-pollinated or crossed among themselves, then the F2 plants with red, pink and white flowers are appeared in the ratio of 1:2:1. Here, the phenotypic ratio matches with the genotypic ratio of monohybrid cross. In F1, red flowers was not completely dominant over white flower. So, flowers appeared of pink color. This is intermediate between the independent expression of the two alleles. The last topic which we are going to discuss is about the codominance. In codominance, both the allelic genes of a genetic trait are equally expressive. That means the dominant character is not able to suppress the recessive character, and thus both the characters they appear side by side in F1 hybrid. For example, as shown in the picture, codominance of coat color in cattle. In cattle, the gene R stands for red coat color and W for the white coat. So when the red coat color cattle are crossed with a white coat color cattle, then the F1 hybrid have a rayon color. The rayon color is the mixture of red and white hairs which develop side by side in F1 heterogeneous organisms. In F2 generation, red, rayon, white cattle are produced in the ratio of 1:2:1. Another example we can take from a plant, when we are going to cross a red colored flower with a white colored flower plant there, then in the F1 generation, we'll be getting a flower which is having both the patches, both the colors, red and white. Here, the red one is the dominant one, but it is not going to suppress the recessive color, that is the white. So, in the F1 generation, for the codominance, we'll be getting a flower which is having both the patches of red and white color. Questions based on the study material.